Welcome. This presentation explores cancer stem cells, where they come from, and why they matter for resistance and relapse. Over the next slides, we'll connect origins, key properties, and experimental approaches to see how CSCs survive therapy and seed new tumors. And we'll highlight potential points for intervention that could improve long-term patient outcomes. This slide summarizes the core traits that let cancer stem cells evade treatment. They divide asymmetrically to self-renew while producing diverse progeny, have heightened DNA repair and ROS detox systems, can enter quiescence to hide from drugs, and activate stemness, signaling axes like WNT or Notch, all of which collectively enable therapy evasion and later tumor reseeding. Identifying CSCs is tricky because common workhorse markers such as CD44, CD133, and ALDH1 overlap with normal tissue, complicating isolation. The slide shows how tumor-specific refinement markers like LGR5, CD24, and CD90 can be combined to enrich CSC populations, and emphasizes that marker panels must be tailored to tumor type to improve yield and reduce contamination from normal cells. Here we outline the methods researchers use to define and validate CSCs. Gold standards include sphere formation, assays for self-renewal, limiting dilution xenografts to test tumor initiation in vivo, and lineage tracing to follow clonal dynamics, while complementary tools like flow cytometry and single-cell transcriptomics map heterogeneity and stem-like states, and integrated data analyses reveal functional hierarchies and vulnerabilities. This diagram shows how tumor-initiating cells evolve. Acquisition of driver mutations, such as KRAS or TP53, and changes in epigenetic modifiers, like DNMT1, produce hierarchical clones. Those clones expand differentially, creating subpopulations with varying malignant potential. And over time, selection favors highly malignant clones that drive progression and make tumors harder to treat. Epithelial to mesenchymal transition regulators like Twist and Snail repress E-cadherin and enable motility, which the figure links to circulating tumor cells that retain stem properties. Those CTCs can survive in the bloodstream, extravasate into distant tissue, and seed metastatic colonies, illustrating how EMT and stemness cooperate to drive dissemination and secondary tumor formation. Dormant CSCs survive chemotherapy through multiple mechanisms. ABC efflux pumps reduce drug accumulation, antioxidant enzymes manage redox balance, and robust DNA damage checkpoints allow repair and pause. Adaptive responses, including enhanced detoxification, oxidative stress control, and temporary cell cycle arrest, enable CSCs to later re-enter the cycle, repopulate tumors, and seed residual disease, leading to relapse. This slide explains how non-stem tumor cells can regain stemness through plasticity. Environmental stresses like hypoxia, TGF-B signaling, or cytotoxic therapy trigger epigenetic reprogramming, leading to re-expression of key factors such as OCT4, SOX2, and NANOG. The result is regained tumorigenicity as cells transition back into a cancer stem cell state, complicating eradication efforts.